students, welcome you back to my channel Biomixer. Hope you are doing well. So in the last two videos we have already discussed about the signaling pathway of auxin and gibberellin. Hope that videos are helpful for you for your preparation and for your examinations. So if you like that videos, please do not forget to like, share and subscribe. So in this today's video we will discuss about another one signaling pathway of a phytohormone, which is a gaseous phytohormone, that is ethylene. So as we are discussing here about the signaling pathway, for a signaling pathway it requires a ligand as well as receptor. In this case the ligand is the ethylene and for that receptor is present, which is known as ETR1. This ETR1, it is a receptor for the ligand ethylene. This ETR1, it is a homodimer and it is an integral membrane protein. It is made up of two integral membrane protein, and these two integral membrane proteins are similar proteins and they are attached together by the help of a bond which is known as disulfide bond. It is made up of disulfide bond. In the first case we will see what happens when there is no ethylene. In the second case we will see what happens in presence of ethylene. So here there is a copper ion which is present, and this is a copper dependent. So the binding of this receptor and the ligand, it is by the copper dependent. So this ETR1 in the C-terminal domain, it is having another one protein which is known as CTR1. And the CTR1, it is a kinase and it is active when there is no ethylene. In absence of ethylene, the CTR1 protein is active. Okay. So what happens when this kinase is active? It will add phosphate. To whom it will add phosphate? It will add phosphate to the EIN2. This EIN2, it is also an integral membrane protein which is present in the endoplasmic reticulum, and its C-terminal portion is present inside the cytoplasm. So what happens? The CTR1, which is a kinase, it phosphorylates the C-terminal domain of EIN2. Okay, EIN2. So here there is phosphorylation. Due to this phosphorylation, here the C-terminal of EIN2 cannot be cleaved. So what was the requirement of this cleavage of this EIN2 C-terminal? Why the C-terminal of the EIN2 needs to be cleaved? Because this inside the nucleus there is another one factor which is known as EIN3. Okay, which is responsible for the transcription of ethylene-responsive factors. Okay, this EIN3, it is a protein which is required for the transcription of the ethylene-responsive gene. So inside the nucleus, this EIN3 is being degraded by 26S proteasome. So for protecting this EIN3 inside the nucleus from degradation, the C-terminal of EIN2 is required. Okay. So in absence of ethylene, this EIN2, C-terminal of EIN2 cannot be cleaved because CTR1 has already phosphorylated it. So this EIN2 cannot be cleaved. So it cannot enter inside the nucleus. As it cannot enter inside the nucleus, the EIN3 is being degraded. So there is no transcription of ethylene response factors. Okay, so ethylene responsive genes are not produced here. So there is no formation of the genes which are responsible for the production of ethylene. Ethylene responsive genes are not produced. Okay, in the next case we will see what happens in presence of ethylene. Here we can see these are the ethylenes which are present, and for binding this ethylene with this receptor, it requires a copper ion. It is a copper dependent mechanism. So in presence of ethylene, what happens? The CTR1, it gets inactivated. Like we can see here that it is being detached. This CTR1 which is present, it is being detached. They are not connected now. Okay, so now they are inactive. As they are inactive now, it was a kinase, CTR1 was a kinase, so it cannot phosphorylate the C-terminal region of EIN2. This EIN2, it was having a C-terminal, so this C-terminal part cannot be phosphorylated. Now the C-terminal is cleaved from here. Okay, what happens if the C-terminal is cleaved? It is being transported inside the nucleus, so the nuclear pores, And what is this? It prevents the EIN3 from 26S proteasomal degradation. Now, what was the work of this EIN3? This EIN3 was responsible for the transcription, for helping for the transcription of ethylene responsive factors. Okay, this is how the regulation of ethylene signaling pathway is taking place in presence as well as in absence of ethylene. So what was the work of ethylene? What happens if there is presence of ethylene or in absence of ethylene? Like here from this diagram we can understand. So this is in presence. Here we can see that there is a triple response in presence of ethylene. Okay, here we can see here the ethylene, where the ethylene is present, the type of stem, here we can see it is short and it is little bit thick. Okay, and it is curdling. There is a curdling. Okay, this is the indication that there is the signaling of ethylene present over here, as well as there is epinastic bending, or we can tell that it is a downward bending in presence of ethylene treatment. When the ethylene is being treated, these are the conditions which we can observe in case of a plant. Okay, this was how the process of ethylene was taking place and how the function of ethylene was taking place. Hope this video will be helpful for you. In the next video, we will discuss the questions which are asked in CSIR Life Science Net. Thank you everyone for watching this video till the end, and if you like this video, please do not forget to like, share and subscribe. Thank you.